The first phase of the bacterial growth curve is the lag phase, and the lag phase is just this little tiny portion right here. It is important to note in the lag phase, the number of bacterial cells, the quantity, is not increasing substantially, so that we don't see a large number of bacterial cells being produced right here. What is important, though, is that this is when these cells are getting used to their new media, they're processing nutrients, and they are producing a large amount of energy, or ATP. So I always think of the A in lag phase for ATP. This is when they're producing energy, because in the next phase, they are going to need that energy in order to sustain a pretty substantial amount of growth. So the next phase is the log phase. The log phase is what we see right here. In the log phase, bacterial cells are going to be growing and dividing at their most rapid rate. So they're going to use all the energy that they made in the lag phase and now use it to drive binary fission cycles. So they will, 2 will divide and become 4, and then 4 will divide and become 8, and 16, and 32, and 64. Until they run out of nutrients. But during the log phase, this is when we see the highest amount of bacterial growth. And then this phase will continue again until the environment becomes unstable, which would typically happen when they run out of food or nutrients. The stationary phase is this top phase right here. And we see it's kind of a flat line. So in the stationary phase, the number of new cells being produced equals the number of older cells that are now starting to die off. The thing you have to remember is at this point, the media within the petri dish has been used substantially. So there's nutrient depletion, and then waste is starting to build up. And because of this, some of those original cells are starting to die off. So yes, we're still producing new cells at this point in the petri dish. We maybe just produced 1,000 new cells, but 1,000 of those original cells that have been in the media longer, they just died because of that waste buildup and nutrient depletion. So the overall number of cells in the petri dish actually remains the same at this point. So during the stationary phase, cell growth equals cell death. Then during the death or decline phase, which is what we see here, during this time, there's no food left. All the nutrients have been depleted. There is toxic waste everywhere. And essentially what happens is all bacterial cells start to die. So we see this rapid decline in the overall number of bacterial cells in the petri dish. This is the point, though, that if a bacterial cell can, it will produce an endospore. If it can't, it just dies. But if it's able to, it is now in a harsh environment where there's no nutrients and the environment's surrounded by toxic waste. So if it's able to, it'll form an endospore and go dormant in hopes that it will at one point get transferred into a more sustainable environment. But again, if it's not able to produce an endospore, it has nothing to protect itself, and it will just essentially die off. So with bacterial cells, often we do like to ask ourselves, if we collect a sample from a surface, how many bacterial cells were present within that original sample? So that is sometimes a question that gets asked. In order to do that, you have to inoculate your plate in a way that you get nothing but isolated colonies. Because in order to figure out how many cells were present in an original sample, you have to dilute your sample. So you do have to know something called a dilution factor. And then you also have to know the number of colonies that exist on a plate. So in order to get colonies on a plate, you do have to do a specific type of inoculation technique. And again, just as a refresher, a colony is a macroscopic mass of cells that has originated from a single cell that has been allowed to grow and divide. So all of these individual circular units are all colonies, and this again is where one bacterial cell was placed 24 hours ago. That bacterial cell continued to grow and divide until it formed this visible macroscopic cluster of cells that we view on a petri dish and identify as a colony. So if you are tasked with the goal or tasked with the project of finding out how many bacterial cells are in an original sample, unfortunately you just can't take a sample of the specimen and then put it on a petri dish, because you'll get something like this where there are no colonies and there was an abundance of bacterial growth, so all the colonies just merged together and made this big blob. Now we do often do things like street plating techniques, but a street plate technique only gives you isolated colonies and 1 to 2 quadrants, so in that quadrant 3 or quadrant 4. With this technique, you need your entire petri dish to be nothing but isolated colonies. So whereas in a street plate, you would dilute out the sample as you went through the different quadrants, with this specific technique, you're actually going to dilute the sample before you put it into the petri dish. So what you do is you take your original sample of whatever you want to know how many cells are present, and you need to dilute it out with water before you put it on the plate, because if we want to know how many bacterial cells are present in here, and we take a sample from this, the original sample, and put it on the plate, we're going to get something like this. We're not going to see any colonies, we're just going to see a big blob of bacterial growth. So essentially what we do is we take some of the original sample and we need to dilute it with sterile water. So typically we'll take a milliliter from this and we'll put it into 9 milliliters of sterile water, and that dilutes it down by 1/10. Then we'll take 1 milliliter of this and dilute it into 9 milliliters of sterile water, and we'll dilute it down by 100. And then we can continue. We can take a milliliter from this, put it into 9 milliliters of sterile water, dilute it by 1,000, and then one last time, a milliliter from this into 9 milliliters of sterile water, and dilute it down by 10,000. By the time you get here, you've diluted down the quantity of bacterial cells substantially, and we can take a sample from this, put it on a petri dish, and get something that looks like this, where there's much less bacteria, so they have the space to mature into isolated colonies. And then the next step that we need to actually do is count the number of colonies on the plate, because in order to figure out how many bacterial cells are present in the original sample, we need to take the number of colonies on the plate and multiply it by the dilution factor. So obviously, step 1 is kind of creating a plate that has colonies that we can count. So there is actually a specific rule as to how many colonies you can have on the plate. So the rule is the plate has to have somewhere between 30 to 300 colonies. If we have less than 30 colonies, it's not ideal. It's very kind of statistically insignificant, and we would like to have a higher quantity. So less than or more than 30 is required, but at the same time, can't have too many. So the rule is if there's more than 300, we do not want to use that plate for a colony count. So as long as the colony count range on the plate is between 30 to 300, we can continue on. So if we look at this plate right here, this plate has way more than 300 colonies. And especially down here, you can see how the colonies are really merging together, so we can't really get a clear count on exactly how many are present. But it's definitely over 300, so this plate, no good. We would have to dilute out the sample more and re-inoculate it to get less colonies, which is what we have here. Right here we have a plate that's been diluted out more, so we have less colonies. There's actually 81 colonies, which fits in between the 30 to 300 range, so we are good there. So to figure out how many cells were present in the original sample, all you have to do is take the number of colonies, which is 81, and multiply it by the dilution factor, which is simply just a number I'm going to give you. So in this case, the number of colonies was 81. The dilution factor was 100,000. That gives us a total of 8,100,000. That tells us that there were 8,100,000 bacterial cells per milliliter in the original sample. The units of measurement that we actually use for this are called CFUs per milliliter. A CFU stands for colony forming unit. So when I say there is 8,100,000 CFUs per milliliter, I'm basically saying there's 8,100,000 bacterial cells per milliliter, because one CFU, one colony forming unit, is equivalent to one bacterial cell. So if we were doing this test and looking at these Petri dishes, so right here we have our original sample, and our goal is to figure out how many bacterial cells are in this original sample. So we'll take a sample of this and we'll dilute it out with water, and then inoculate those samples onto a Petri dish to figure out which one's the best to get our colony count, and then multiply that by the dilution factor. So we wouldn't want to use this plate. If we look at this plate, it's really hard to get a colony count because we can see where there's a lot of colonies merging together, possibly over 300. Same with this one. This area here, we really can't get a good colony count, so we wouldn't want to use that one. These two here, it's quite easy to get a good colony count. However, we do not want to use this one right here. I know looking at it, it looks ideal, but there's definitely less than 30, so that doesn't fall into our 30 to 300 colony count range, so we cannot use that one. This one right here, though, is ideal. There's actually 43 colonies on that plate, so if we take the number of colonies, 43, multiply that by the dilution factor, which is 1,000, that gives us an answer of 43,000. That is 43,000 CFUs per milliliter, or 43,000 bacterial cells per milliliter were present in that original sample. And the last thing we'll talk about is turbidity. When we put bacteria in a Petri dish, we can obviously see them growing on top of the solid surface as a colony, but when you put them in a liquid, you don't see colonies. Instead, you see cloudiness, or what we call turbidity. So typically, when we work with nutrient broth, if there's no bacteria in it, it is going to be clear as what we would see here. Once you add bacteria to the broth and they start to grow, that solution becomes cloudy, which is what we see here, and that cloudiness is known as turbidity. And typically, the more cloudy or the more turbid the solution is, the more bacteria are present. So we want to think of turbidity as having bacterial growth. And typically, again, the more turbid it is, the larger the population size.